Okay, so what if I told you that we can produce life-saving medicine, like insulin, inside one of the simplest little organisms on the entire planet? It sounds wild, right? Well, today we're diving into exactly how scientists have turned tiny bacteria into these incredible, powerful medicine factories. And hey, if you're studying biology, this is a core concept you are absolutely going to want to master. Let's get right into it. So here's our game plan. We'll start with why this was a problem in the first place, the problem of supply. Then, we'll open up biology's molecular toolkit and meet the key players. After that, we're walking through the actual procedure, step by step. We'll see how we go from a single bacteria to mass-produced medicine. And finally, we'll zoom out to see the huge potential of what we call transgenics. All right, so first things first, let's set the stage here. Why do we even need to do any of this? Well, think about diseases like diabetes. Millions and millions of people need a constant, reliable supply of the protein insulin just to live. And historically, getting enough of it was a massive, massive challenge. We desperately needed a better, more efficient way to make it. And that leads us to this really fascinating biological puzzle. How on earth can a simple little bacterium be told to make a complex human protein? I mean, it seems like it should be impossible, right? But the solution to this is actually one of the biggest breakthroughs in all of modern biology. And the answer? The big idea here is a field called genetic engineering. All it really is, at its core, is purposefully editing an organism's DNA. And what's the goal? To give it a new set of instructions, to tell it to build a specific protein, which then gives it a brand new trait, like, say, the ability to produce human insulin. So, to really get how genetic engineering works, we first need to get familiar with the tools we're going to be using. You can think of it like a biological workshop, right? But instead of hammers and wrenches, we've got this highly specialized molecular toolkit. So, let's meet the key players. Our little factory floor is, of course, the bacterium itself. Now, if you look inside, you'll see its main DNA, that big circular chromosome. But the real star of the show, for our purposes, are those smaller, separate loops of DNA you see there. These little loops are called plasmids. Now, what's so important about them is that they exist separately from the main bacterial DNA. Plasmids often carry these kind of bonus genes, things like antibiotic resistance, and this is the crucial part, they can be easily passed from one bacterium to another. This ability to be transferred makes them the absolute perfect vehicle for delivering our new genetic instructions. But you might be wondering, why can a bacterium even read a human gene in the first place? Well, it all comes down to the fact that all life on Earth shares a universal operating system. Seriously. Whether you're a tiny bacterium or a person, you use the exact same DNA and RNA building blocks, the same 20 amino acids, and the same protein-building machines called ribosomes. It's like all life is running on the same basic software. Okay, now that we've met our players, let's get to the main event, the procedure itself. And honestly, the easiest way to wrap your head around this whole thing is to think of it as a simple cut-and-paste job, just happening on a molecular scale. Now, this process has a formal name, and you're going to want to remember this one for your exam. It's called bacterial transformation. That's the official term for this whole process of giving bacteria foreign DNA so they can churn out foreign proteins. We are quite literally transforming what that bacterium can do. So how do we do the cut part of our cut and paste? We use a super special tool called a restriction enzyme. You can think of it as a pair of molecular scissors, and these scissors are programmed to snip DNA only at a very specific sequence of code. It makes this special staggered cut, which creates what scientists call sticky ends on both the human gene we want and the bacterial plasmid we're putting it into. And when we paste that human gene into the open plasmid, thanks to those sticky ends, we create something totally new. This is called recombinant DNA, and it's exactly what it sounds like. A hybrid molecule. It's a combination of DNA from two completely different species, part human, part bacteria, all rolled into one. So let's put it all together. It's basically a 5-step process. Step 1, you get the human insulin gene. Step 2, you get a plasmid out of a bacterium. Step 3, you use the exact same restriction enzyme, our molecular scissors, to cut both of them. Step 4, you use a different enzyme called ligase, which acts like our molecular glue, to seal the gene into the plasmid. And finally, step 5, you put that new recombinant plasmid back into a host bacterium and just let them reproduce. So we did it. We successfully transformed one single bacterium. That's great and all, but how does that solve our original supply problem? Well, this is where we scale things way, way up.
moving from a single cell to a full-blown industrial factory.
And here you can see the whole process from start to finish. The recombinant DNA we made gets put into a host bacterium. Then, that one special cell is placed in a giant fermentation tank. It's basically the perfect environment for bacteria to grow and multiply. And as they reproduce by the billions, every single new cell is a perfect clone that carries our instructions, acting as a tiny factory just pumping out human insulin.
So what's the end result of all this incredible science? It's a cheaper, faster, and much more reliable way to produce pure human insulin. This technology completely revolutionized the treatment of diabetes. It's made these essential medicines accessible to people all over the world. This is why this stuff matters so much.
Now, making insulin is just one, absolutely incredible application of this technology. But let's zoom out for a second and look at the bigger picture, because this whole cut and paste technique has opened up a whole new field of biology. Our little insulin-making bacterium? It's a perfect example of what we call a transgenic organism. And that's another key term for you. It's the name for any organism that has been modified to contain genes from a different species. And it's definitely not just limited to bacteria. I mean, the applications here are just incredibly broad. Beyond making medicines, genetic engineering is used to create things like pest-resistant crops to help secure our food supply. It's used to develop organisms with all sorts of desirable traits. It's fundamentally changing the game in both agriculture and medicine. So there you have it. We've gone from a huge medical problem to the molecular toolkit, we've walked through the procedure, and we've seen how it scales up to change the world. So now that you understand the basic cut and paste power of genetic engineering, what kind of real-world problem would you want to solve with it? It's an incredibly powerful tool, and the possibilities are pretty fascinating to think about.